Now let's discuss about the specificity of the enzyme. Now we know that enzymes are protein and hence they have a specific three-dimensional conformation as a result of the unique twisting and folding of polypeptide chains. And this unique twisting and folding of polypeptide chain form active sites on the enzyme. Therefore, enzymes are highly specific. Now, what do you mean by specific? Well, specificity is the ability of an enzyme to bind to a particular substrate only, and thus it will only be able to catalyze a particular substrate. So this specificity, it operates through the conformational and structural complementarity between the enzyme and substrate. Based on that, various enzyme specificities are given. Actually, there are main three specificity of enzyme. One is already given here, substrate enzyme specificity, then stereospecificity or optical specificity, and reaction specificity, which is of two types, that is bond and group specificity. So we will discuss one by one all these different types of specificity: bond specificity, group specificity, substrate, optical, geometrical and cofactor specificity. First we will see what do you mean by bond specificity. Here in this case, enzymes, they catalyze only those substrates which are having similar bonds and similar structures. This specificity is comparatively a very less type of specificity, and it is also called as relative specificity. Now in this diagram, a very good example is given, peptidase. This enzyme, peptidase, it can catalyze only peptide bond. It can catalyze the compounds if the peptide bonds are there. No matter which groups are attached here, whether two glycines are attached or two alanine or valine are attached, peptidase, it is related to only bond. If peptide bond is there, it can catalyze that substrate. Such specificity is called as a bond specificity. Another example is amylase. Amylase, it can catalyze only alpha 1,4 glycosidic linkage. That means it is related to only alpha bond. It cannot catalyze beta 1,4 glycosidic linkage. Lipase, this enzyme can hydrolyze only ester bond. If ester bond is present between glycerol and fatty acid, then only it can catalyze those lipids. So, peptidase, amylase, lipase, these enzymes shows bond specificity because their catalytic activity is based on the bonds. Another is group specificity. We just saw that enzymes are very specific to bonds. At the same time, they are very specific to the group surrounding to it. This specificity is quite more than the bond specificity. It is also known as moderate specificity as well as structural specificity. Best example of this is pepsin. This enzyme, it hydrolyzes peptide bond. But which peptide bond? Only those peptide bonds in which the amino group has aromatic amino acids, or amino group is contributed by an aromatic amino acid. We know that out of 20 amino acids, there are some amino acid which has aromatic ring in their structure, like phenylalanine, tyrosine, tryptophan. So pepsin, this enzyme can catalyze only those bonds. You can see the picture of scissor. It is near to phenylalanine, tyrosine and tryptophan because these are aromatic amino acids. Pepsin cannot cleave this bond because the bond is between glycine and methionine and both are not aromatic amino acids. So some amino acids, they are very specific about the groups, that if aromatic group is there, then only they can catalyze the reaction. Such specificity is called as group specificity. Next example is optical specificity, which is also called as stereospecificity of the enzyme. Just now I told you that alpha amylase, it can catalyze only alpha 1,4 glycosidic linkage, but it cannot catalyze beta glycosidic linkage. Alpha and beta, they are just stereoisomers of each other. But alpha amylase, it can catalyze only alpha form and not the beta form. Another example of this you can see here. See here, this blue part is the enzyme part, okay, and here this is a substrate. This substrate, in this substrate, this is amino acid and it is L amino acid because NH3 is on left side and H is on right side. Here the structure is same. You can see the amino acid structure is same. Only difference is here H is on left side, NH3 plus is on right side. So this is L-alanine and this is D-alanine. The enzyme whose name is L-alanine oxidase, it can catalyze L-alanine, but L-alanine oxidase cannot oxidize. You can see this cross. L-alanine oxidase cannot oxidize the D-alanine. So this is a stereospecific enzyme. Now geometric specificity of the enzyme. One thing I just want to make it clear that names are changed. This is methanol and this is ethanol. Only this difference is there. Okay, it's mistake. Now here, what is geometric specificity of the enzyme? Some enzyme, they catalyze those substrate which has some geometric part similar. Now you can see methanol has this CH2OH group as well as ethanol also has CH2OH group in its structure, in its geometry. So this is the enzyme, blue part. Alcohol dehydrogenase, this is the enzyme, it can catalyze even on methanol as well as ethanol. Why? Because their geometry is somewhat same. Such specificity is a geometric specificity. While substrate specificity, in this type of specificity, the enzyme acts only on one type of substrate. For example, urease. Urease, it acts only on uric acid, while maltase, it acts only on maltose. So such type of specificity is known as substrate specificity. Now the last type of specificity is cofactor specificity. But before starting, what do you mean by cofactor specificity? Let's see. What do you mean by first? What do you mean by cofactors? Now, see here, we know that many enzymes require certain non-protein small additional factors for their catalytic activity. Now see here, the holoenzyme or the whole enzyme, complete enzyme. This is the whole enzyme. Holoenzyme, it is made up of two parts, that is apoenzyme. Apoenzyme is a protein part, but it is inactive. But when apoenzyme combines with coenzyme, coenzyme, it is a part of cofactor, and cofactor is nothing but a non-protein part, which is also called as activator. So when this apoenzyme combines with coenzyme, there is formation of holoenzyme, and holoenzyme, it is very active and it can combine with the substrate and start the catalytic process. In this example also or diagram also, you can very well understand what do you mean by apoenzyme, coenzyme. Now see, this is the whole enzyme, this complete, which is called as holoenzyme. Holoenzyme is made up of apoenzyme, which is a protein part, but it is alone, it is inactive. Now in this apoenzyme there is one more part, that is non-protein part, it is a coenzyme, and along with coenzyme one more non-protein part is there, that is called as cofactor. And when all these come together, enzyme becomes active. Now it has this catalytic site or active site where it can start its catalytic activity. So one thing is clear, that enzyme, it is made up of two parts, apoenzyme and coenzyme. Coenzyme, it is nothing but it is a part of cofactor. Now let's see what do you mean by coenzyme and cofactor, or what is the difference between these two. As I already told you, some enzymes, they alone cannot act. They need some activators or they need some helpers. There are two helpers or two activators which help the enzymes for their catalytic activity. One is cofactor and another is coenzyme. Now both are, cofactors and coenzyme, both are non-protein part. Cofactors, these are the small inorganic compounds or ions, while coenzymes are organic molecules. These are organic molecules, not ions. Now cofactors, they bound with the enzyme molecule, while coenzyme also bound with the enzyme near the active site. Some metal ions like Mg, Ka, calcium ion, zinc, iron, these are the examples of cofactors. Here you can see two examples are given. Iron in hemoglobin, it acts as a cofactor, as well as magnesium in chlorophyll, it also acts as a cofactor. Now what do you mean by coenzyme? Actually about coenzyme we have already discussed in the vitamin chapter. Almost all vitamins, they have coenzyme parts. So coenzymes are again non-protein, but these are the organic molecules and they bind either temporarily or permanently to the enzyme near active site. Like NAD is the coenzyme which present in vitamin niacin or B3, FAD which is along with riboflavin, while coenzyme A, it is again an example of coenzyme. Here some list of coenzyme and cofactors and the enzyme to whom they help. For example, thiamine pyrophosphate, this coenzyme help the enzyme pyruvate dehydrogenase. Flavin adenine nucleotide FAN coenzyme help monoamine oxidase. NAD, it helps lactate dehydrogenase. Pyridoxal phosphate, it helps glycogen phosphorylase, and so on. While these are the cofactors, these metal ions, they help these enzymes for their action. Now here I have listed some difference between coenzyme and cofactors. Actually both are related with each other. Coenzyme, it is a part of cofactor. So coenzyme is a small organic non-protein molecule that carry chemical group between enzymes, while cofactor is also a non-protein, but it is mostly inorganic compound or metal ion that tightly or loosely bind with an enzyme or other protein molecule. Coenzyme, it is a type of cofactor, while cofactor, they are of two types, like coenzyme and a prosthetic group. Coenzymes, these are organic molecules. These are organic molecules, while cofactors, these are chemical compounds and these are inorganic chemical compounds. Coenzymes are mostly loosely bonded to the enzyme, but cofactors sometimes covalently bonded to an enzyme. Coenzyme assist biological transformation, while cofactor help for the catalytic activity of the enzyme. Coenzyme, they serve as carrier to the enzyme, while cofactor, as just told you, that it increases the rate of catalytic reaction. As coenzymes, they are loosely bonded, they can be easily removed from the enzyme, while cofactors can be removed from the enzyme by denaturing process. Examples of coenzyme, here these are listed: TPP, FAN, acetyl coenzyme, coenzyme A, biotin, etc. While metal ions like Zn2+, Mn2+, K+, Mg2+, all these are the examples of cofactors. Here we have finished the complete chapter enzyme. Thank you so much.